3 times dividing by mitosis to produce that cell with 8 haploid nuclei. So yeah, 6 and 9 were really the only ones that seemed to kind of get a majority of the students. But are there any y'all want to kind of go through now before we move on to lecture?
Are you able to give us any point for like the last two?
For 9 and 10?
No, like the most difficult ones, like 6
Oh,
or 9.
no, because I've been going by that rule of, if basically a majority give another answer, then it shows that, it's a poorly worded question.
Okay,
But there weren't any like that this time around. This is, I'll say this one, although actually this semester might break that chain, but this one is usually the hardest one of the IRATs. I just looked briefly. It didn't look like it was actually all that lower than previous IRATs, but
Wait a minute.
in other semesters it's a lot lower.
Okay, I wrote down what the video said. And
You-
it said
you wrote down the
it
video?
said it says the female gametophyte is the the development of the female gametophyte is the division of the megasporocyte by meiosis. Inside the ovule, the megasporocyte 2N undergoes meiosis to form 4 cells, but only one of these cells survives as the megaspore N.
So the gametophyte will form a haploid megasporocyte. So the megasporocyte at this point is haploid.
But it says
Is that a note? Well,
no. In the video, it said it. I wrote down what the subtitles in the video.
Then.
Can we Google it really?
Wait, the subtitles
In the video.
So don't go by the subtitles because the subtitles
Can you just Google it?
usually screw it up.
It literally says meiosis of megasporocyte.
I'm corrected.
Yay!
Yeah, okay.
Yeah.
So we're getting a full point here. Good. Yeah. Megasporocyte is a diploid cell in plants in which meiosis will occur, resulting in the production of 4 haploid megaspores. And then they will divide. Good.
Boom.
Okay, +1.
Oh, yeah.
I like
Boom.
that. Thank you.
So question 9
Good job, Hava. Megasporocyte does indeed divide by meiosis, a total of, it's actually just a poorly written question all in all, because it only undergoes meiosis once, and from there those cells will undergo mitosis. So this is a bad question.
Yay!
Breaking through. Thank you, Hava.
So that's automatic 10 points for everyone?
An automatic point, if you're thinking in terms of 100%, automatic 10%.
Yeah.
Yeah. Good. Oh, good catch. Nice. All right. I love this. Okay, so, any other questions y'all want to go through though? Maybe this is all wrong.
So you're telling me instead of getting a 90, I got a 10?
Well, so we have to take from a pool of 1302 questions. So basically this just means, I took from the pool and we have to get rid of this question. All right. I wonder
Or
how
change
many
it.
past classes
They have to
not-
change the question.
I just
answer.
remove it from the pool and then replace it with a different megasporocyte
Oh,
question.
okay.
Yeah. Well, I'll have the people in charge of 1302 do that.
He'll make a complaint and tell him.
Not even a complaint. I'm just going to tell them it's wrong. Nice. Okay. Any other questions that y'all want to go through? No?
Oh wait, can you explain the cycle thingy, the 14-day thingy?
Which one?
The go up. Unless you're gonna lecture about it. Are you gonna lecture about the menstrual cycle?
Yeah, the plan for the rest of Module 5 to try and catch up is today we're going to talk about plant reproduction, and then we've only got 2 more Module 5 days left. So Monday will be female reproduction in animals, and then Wednesday will be male reproduction, and then ending with the concept check quiz. So yeah, a whole lot of the female reproduction aspect is really just kind of getting the hormones and anatomy involved in really this 28-day menstrual cycle down, essentially. It's a lot of that on the test, so I try to kind of focus primarily on that. And so we will go into like a couple of activities that are just fleshing this out. But yeah, for these plants we covered today though. But yeah, is there anything specific you do want to kind of cover? We'll talk about 4 in detail. We're going to come back to this question on Monday if that was a difficult one. The rest of these we will also, this we won't kind of bring up again, but not many people got this wrong. Human sperm and eggs have the same number of chromosomes. Eggs are much larger than individual sperm cells. Sperm have the flagella that allow for mobility, eggs do not. And sperm are produced from puberty until death, but eggs are not. So that's really the only one. The rest of these we will kind of go in detail on Monday. All right, any questions? Okay, cool. Then what we will do is not talk about reproduction. I just want to kind of spend a few minutes talking about one concept we really didn't talk a lot about because of the missed class, but you definitely want to be aware of for the last exam. So unit exam 3 is the last time we meet, actually, is unit exam 3. And let me go ahead and pull these slides up. So before, the last time we met, we kind of focused on circulatory systems. And you really want to get kind of the flow, imagining you're a red blood cell starting at point A, you want to know what all you visit and in what order. In 1102, I kind of talked about, and I mentioned it briefly, but one way to kind of think about it, again, this is just a generalization. It's not naming any specific structures, but when you're thinking of these veins, atria, arteries, ventricles, think of, V goes to A, goes to V, goes to A. It kind of goes back and forth. So say you're starting at a vein like the vena cava, which is one of those veins. A vein is going to go into an atrium. From the atrium, you're going to enter a ventricle. From a ventricle, you're going to leave the heart through an artery. Eventually, an artery will go wherever it needs to go. Say you were starting, at the vena cava, you're at the right atrium, right ventricle, pulmonary artery, which goes to the, it rhymes with dung, lung. Yeah. But, after you go wherever you need to go, the artery is going to kind of lead back to another vein, like the pulmonary vein, which now goes through the left atrium, the left ventricle, another artery, the main artery, aorta, which goes wherever the oxygen was needed, coming back through a vein, back to the vena cava. So it's just this process of V, A, V, A, V, A, V, A. This doesn't tell you, what the vena cava is. It doesn't tell you right atrium versus left atrium, but at least, it's something to start off with, something that you can maybe build upon from there. So we got some practice with circulatory systems last time. What we'll kind of do for the first little chunk today is talk about the sort of analog in plants. Obviously plants don't have blood, but they have a faux circulatory system, something that transports needed stuff throughout the plant. And so in plants, this is referred to as transportation. And the way it travels is actually not very circular, hence it's not called the circulatory system. I just call it the faux circulatory system, because it's really not circular travel. It's kind of one way into the plant and a different exit out. And the way we describe this is roots to shoots. So from the roots, water kind of begins its travel up the plant until it leaves through the leaves. That's why they're called leaves. No, that's not why they're called leaves. But so, you know, there are a couple of kind of vocab words that get kind of hidden within this topic of plant transportation. One of them sounds just like transportation, but it's called transpiration. So transpiration is different than transportation. Transportation is the whole process, just the water traveling from the bottom to the top. How that happens is through transpiration. So transpiration is essentially the equivalent of plant sweat. So when plants are heated up, water evaporates from their leaves, and this whole process essentially creates a continuous vacuum that keeps water climbing up and up the plant. And so this is essentially how plants are able to move water and xylem sap kind of potentially long distances from the very bottom, those roots, all the way up. It's through this process of transpiration. And I just kind of want to walk through, because your book kind of shows you pretty complicated diagrams, but the process that's really going on is pretty simple overall. You know, it's got these, in this one diagram, in 2918, we see like 5 different points they're making. But really, the main things to know about this whole process are 2 chemical properties that are just chemical properties of water that drive this whole process. And those are cohesion and adhesion. So cohesion is water sticking to water. Adhesion is water sticking to something else. The walls of, you know, plant cell walls, xylem cells that allow kind of the water to actually climb. And if you're having trouble thinking between, you know, of the differences between the two, just think of where you've kind of heard versions of these words before. When something adheres to something, you know, you might be thinking of maybe tape or a sticker or something, right? Those things have good adhesion. It's a sticker sticking onto something that isn't a sticker. It's tape sticking onto something that isn't tape. So adhesion is one thing sticking to something else. In this case, it's water sticking to something that isn't water. Cohesion, you know, the word you might have thought of is, you know, what makes a cohesive group, where each member works well together. Cohesion is the same process here. It's water sticking, working well with water. So cohesion is the fact that water sticks to water. Adhesion is water sticking to something else, in this case, the xylem cells. So water is good at both. It's good at sticking to itself through hydrogen bonds, and it's good at sticking to other things through those same hydrogen bonds. And so based on those two properties, what happens is you have, for example, a plant. This will be, you know, my basic plant that for some reason is just one leaf long. And it's got some root system. And so basically what happens is water is going to fill up this plant just through the fact that nature you might have heard the phrase, nature abhors a vacuum, nature hates a vacuum. So it's a process that we probably see all the time, right? So whenever, for example, you have, I don't know if y'all have seen these kind of puzzles before, but there might be a cup here and another cup here. And there might be the question, what happens when you kind of open this kind of gap in the water? It's going to fall down, right? It's going to occupy that emptier space. Water is constantly trying to go from, just like with diffusion, where oxygen goes from where there's a lot to where there's a little. Water does the same thing too. It's going to go wherever there is this vacuum gap. And so once there is water throughout the plant, what happens when transpiration occurs? Remember, transpiration, that sweating of the leaves, evaporation of a water molecule. So what happens is there are these tiny holes within leaves, and what do we call those holes? Can anyone remember? Yeah, stomata. Yeah, so a stomata is basically a microscopic hole, of which there are many throughout this leaf. So what happens is, you can imagine there's essentially a water molecule on deck there. And as evaporation occurs, it's just that water kind of evaporating out to the atmosphere. So this area inside the leaf, relative to what's going on outside, is a whole lot of water into an area of the atmosphere where there's virtually none there. So the water escapes, and this just creates a gap. Now, that water molecule was right there on deck, now it's empty. And so what happens? The next water molecule takes its place. And so that creates a gap behind it. So the next water molecule takes that place. It essentially is just kind of like pulling up a rope. Think of that with water too. The adhesion of water kind of helps pull that next water into that empty space. And so it just starts this chain reaction that works its way all the way down until you get to the root tip, which if we kind of zoom in on, we can ask what's happening here. So, if this were kind of that root tip is going to be right in the soil, right? So what happens is maybe you have a root tip here, and then finally the last kind of member of this chain of water molecules was here, and now it gets pulled through adhesion here. So it moves to this next step of the root tip, and now there's an empty space here. Well, what's going to happen? Yeah, some water from the soil is going to enter. So long as soil is, well hydrated, this process can keep going over and over continuously. It's when the soil gets totally dehydrated that you run into a problem, and that's why the plant will start to wither up. It's no longer plump with water. It's kind of reaching the end of the rope, you could say. And so this vacuum kind of starts to fold in on itself, and the plant overall looks withered because there's no water to kind of fill up that space. But this whole process, this kind of rope pulling that water kind of goes through, is just the whole process of plant transportation. It's what allows the motor that kind of is pulling on the rope in the first place is just transpiration. Evaporation of water leaving the leaf up high at the plant, at the highest points of the plant, and it starts pulling the rope. These holes, these stomata, don't exist way down along the stem or within the roots themselves. And so the fact that they only occur towards the top is what kickstarts the whole transpiration process. And so, can anyone remember what this hypothesis was called?
The cohesion.
Wait, what?
The cohesion tension hypothesis?
Yeah, cohesion tension hypothesis. Yeah, so it relies on cohesion and adhesion. So those are the properties, but because scientists are cynical and sadistic, they decided instead to just call it cohesion tension hypothesis. the cohesion of water kind of working cohesively, sticking to one another, and the tension you can think of as the tension of pulling on that rope, pulling on that water chain, working its way up. Why isn't it called the adhesion-cohesion hypothesis? I don't know. But yeah, tension or cohesion tension hypothesis, which relies on cohesion and adhesion. And we're not going to do that word association one. But yeah, this is essentially just drawn out here. The ascent up kind of the whole length of the plant itself is basically, think of these water molecules all connected to each other through those hydrogen bonds, that cohesion, and then the tension that forms between those bonds kind of pulls each other up and up and up. So it all starts with transpiration though, and it only ends when there's no longer any water left in that soil. So that's all I wanted to kind of say. I just wanted y'all to kind of be aware of those vocab terms: cohesion, adhesion, the anatomical structures that are involved, stomata, xylem, transpiration versus transportation. So transportation is the process itself. Transpiration is kind of like the motor that starts everything going. It's that evaporation itself. So any questions about that? That's sort of the last of like circulation stuff before we move on to reproduction.
I have a question.
Yeah.
It's like slightly unrelated, but to transpire means like to occur. So I'm not really sure why. How did scientists decide to use that as like sweating? Because perspiration for a human, so something.
Yeah.
You know?
Yeah, I'm actually not totally sure. This area admittedly is not like my strongest suit when it comes to like the etymology of some of these hypotheses and the vocab terms.
Yeah.
But you're right, it doesn't really make kind of intuitive sense like that to me. Like what transpires is what happened.
Yeah.
But transpiration, I'm not really sure. I can look it up and I'll make a note of it actually. And then maybe on Monday, if I find an answer, I'll see if there's a logical way that makes sense. But yeah, it doesn't really make sense to me either, the etymology of that word. Good question though, yeah. Because yeah, that was one of the, I'm always looking for ways to kind of memorize these things to make, so I'm not just memorizing a bunch of words, but that's one that never really sat well. Any other questions? All right, then if not, we can say goodbye to those lecture slides and move on instead to plant reproduction. Oh, iClicker, yes. I've got iClicker on. I'm gonna try to incorporate this a lot more, just to kind of get your brain constantly thinking of potential exam and then also final exam questions. Log in real quick. Pull up the slides. Alright. Okay, so yeah, we're talking today about reproduction, but just plants. So we'll move on to animal reproduction next week, but today we're going to try and cover everything that you want to know. All of it is kind of based on the alternation of generations. And a lot of it is specific to angiosperms. So if you remember those flowering plants from module whatever, 4 or 3 or something, and we're not looking at explaining all of plant reproduction, but really just those flowering plants in particular for the purposes of this class. They're all kind of built off of each other. They've evolved from gymnosperm reproductive cycles and then more ancestral forms. But we just, for quote unquote simplicity, we look at angiosperms. Here I just highlight this. This is mentioned in your textbook. One of the ways plants reproduce. Part of what made angiosperms in particular so successful is that they no longer relied on just water or wind to pollinate and reproduce. They kind of took advantage of the living cars around them, animal life. And so one of kind of the examples here are burrs. So burdock fruits kind of have these huge hooks on them, which can often kind of cling to animals, and then it'll go as long as that animal goes. So it can go potentially much further than traveling via wind or water, especially if you're a particularly big fruit or seed here. But I just highlight this because this is this is called a burdock fruit. Maybe you've heard of it, maybe you haven't. But the hook of this, even though you might not have heard of it, you've benefited from this hook, countless times. This is what led to the invention of Velcro itself. So sometimes the invention of potentially huge, items, part of everyday use, are based off of some of these kind of simple biological features. We'll skip that. So we'll kind of just start with an iClicker question. So I'll give y'all some time to log on. But it's kind of harking back to that alternation of generations. Let's see how well we remember the alternation of generations, because you're going to want to remember it again for the next exam. But different aspects of it. I'll explain. So blank sporophytes make haploid blanks by blank.
Stop looking at my answers.
Blank sporophytes make haploid blanks by blank. Okay, give you about 10 more seconds to finalize your answers. Speak now or forever hold your peace.
Wait. I chose the wrong one.
All right, finalize answers. And let's see.
Oopsies.
Ba-bang! Overwhelming support for A. You got it, dude. Yeah, so diploid sporophytes make haploid spores by meiosis. So if you remember that sporophytes, whether they're diploid or haploid, that can rule out, either 2 or 3 of those, but hopefully you at least knew that they're diploid, and you can rule out B and D. And then it comes, essentially this game of, spore versus gamete, meiosis versus mitosis. Anything going from more chromosomes, like diploid, to fewer, it has to undergo meiosis. Meiosis is more to fewer. Mitosis, you always end up with the same. Whether you're diploid to diploid, that's mitosis. Haploid to haploid, that's mitosis. It's just diploid to haploid is meiosis. And then just the difference between spores and gametes, which often gets many of us, but part of what we'll do today is kind of get into really the details of one part of this alternation of generations, and hopefully we'll be able to tell the difference apart between spores and gametes. All right, so yeah, this kind of brings up kind of ghosts of Christmas past, this idea of alternation of generations. You should remember that plant life cycles are characterized by what we mean this alternation of generations is going back and forth between haploid and diploid, and then back to haploid over and over again, hence the term cycle for life cycle. You remember that sporophytes are diploid. They produce haploid spores by meiosis. And then what will happen is those spores are going to divide by mitosis to make gametophytes. So the spore, you can almost think of as that baby that's going to become a gametophyte in the haploid generation. And then the diploid version is maybe easier to grasp, a zygote that becomes a sporophyte, the plant we actually imagine in our heads. And it starts off with a zygote. Just like in animals, we start off at the zygote phase and become adults. The only difference is with plants, it's kind of this two-pronged phase. There's the diploid version of that and the haploid version of that. And so for haploids, it's spore growing up to be a gametophyte. All right, another question here. Get the brain going. So haploid male and female blanks produce haploid blanks by blank. Also made these questions 20 minutes before class, and as I read them out loud, I'm like, oh, where's the typo? Like, where's the error that I made? But we'll go through this together. These iClicker questions are new, so if we screw up, we screw up. Well, I screw up. All right, finalize your answers. And let's see. Boom. Overwhelming support for haploid male and female gametophytes produce, oops, I didn't even underline the answer, but this is correct. The majority of y'all got it. So first of all, you can, even if you look at the last one, you might be able to eliminate two right off the bat, because you see haploid making haploid. I just said, whenever we stay at the same ploidy level, it's mitosis. So we can rule out C and D right off the bat. And then it comes down to, the first option, sporophyte and gametophyte. You might recall that sporophytes are diploid, gametophytes are haploid. So sporophyte you might be able to rule out. In fact, none of y'all put that one as the answer, so good on y'all there. B is the correct answer here, even though I didn't underline it. Male and female gametophytes produce those haploid gametes. Just reiterating that point here. They produce specifically single-celled gametes by mitosis. We have known for years, hopefully all of us, what these gametes are called. I've referred to them as male and female gametophytes, and they're each making a male and female specific gamete. We know them more familiarly as sperm and eggs. It's the same terminology for plants, the sperm and egg cells. And what will happen from there is a sperm will find an egg, hopefully, and if it does, they fuse during this process of fertilization to make the zygote. And then we're back at the diploid phase. Zygotes dividing by mitosis, growing up to be that sporophyte. So the cycle continues there. We want to essentially recall the general structure of this alternation of generations. So think back to that activity that I had y'all do with the cards, ordering them in more or less the right way. Well, if we kind of think of that, most generally, that would be I'm just gonna put a line here depicting this transition from some haploid stage to a diploid stage. We know the beginning of the diploid stage is going to be, what is that called? What's going to be the first diploid in a plant? It's going to grow up to be a sporophyte. Yeah, zygote. So that first diploid is the zygote. It's 2N, it's diploid. It's an egg and a sperm fusing to form a zygote. It's going to grow up, it's going to keep dividing over time, staying diploid throughout this process. So it's undergoing mitosis, and it's growing up to be a beautiful plant. I'm blanking on my plant drawing skills, so I'm just going to draw what that plant that we imagine in our head is called, which is that sporophyte. Eventually, that sporophyte is going to flower in some way. And it's within this flower that we're going to get to this transition back to a haploid level through what process? Yeah? Yeah, meiosis is going to be what gets us over the hump, over this division. Meiosis. And what is it going to produce through meiosis? What does a sporophyte make? Spores, yeah. So it's going to make a spore. A spore of some sort. What does the spore grow up to become? The gametophyte. Now, this is where we're really going to focus in the process of reproduction. We're going to really focus, not care too much about kind of this zygote to sporophyte phase, but we're really going to be focusing on, the meiosis aspect, the spore, and the fact that it can become, like the previous question said, a male or female gametophyte indicates actually that there are male and female spores. And this is actually a vocab term that might have been on the IRAT that most people got. It's a vocab term referring to this two-spore, heterosporous. The fact that seed plants will form two types of spores. You can think of them as the male spore and the female spore. Another way to think about it, because it's in a lot of these vocab terms you're going to read about, are mega versus micro. Microspore, megaspore. We already saw the megasporocyte. There's a microsporocyte as well. We kind of go down this trajectory of being either a male or female spore, and depending on the trajectory you go, you're going to make either a male gametophyte or a female gametophyte. And eventually, what does the gametophyte make? Just like the sporophyte makes spores, the gametophyte makes gametes. Yeah, and we already know the names of the male and the female gametes. What's the male gamete? Sperm. And the female gamete? Egg. And then they're going to fertilize, going right back to here. This is essentially what y'all had to know for Module 4 pretty much. For Module 5, we're going to throw vocab terms into this structure that you've hopefully built in your head already with Module 4. The good news is, it's going to be a lot of heavy details right here and overlooking some other parts of the alternation of generations. But that's going to be part of the goal for today. In angiosperms, we see this alternation of generations, and one thing we kind of saw throughout the evolution of plants is, the first plants were gametophyte dominant. Those were the mosses, largely haploid. And we eventually the gametophyte kept getting smaller and smaller until we ended up at the most recent, the newest kids on the block, the angiosperms, the flowering plants, where the gametophyte stage, as we'll see, is going to be reduced essentially to only a few cells. And they totally depend on the sporophyte. So the sporophyte is really the majority of a flowering plant. So this haploid phase that we're going to kind of get into the details of is really just a minuscule portion in terms of size, in terms of number of cells. And so, you know, sometimes life cycles are kind of described by 4 Fs in the animal kingdom, but in the plant kingdom, it's essentially 3 Fs, where we'll talk about each of, really 2 of these more, flowers and double fertilization. Fruits are talked about extensively in your textbook. My kind of hint to you at this point is on exams and finals, we really don't ask you all that much about, you know, the fruit process. All those vocab terms, you really, if you're prioritizing your studying time, focus kind of on these aspects up until the fertilization. You know, the fruit part, it's good to know, but that's my kind of hint there. But yeah, so a lot of the important stuff that happens, that gets to fertilization, happens in the reproductive organ of a plant, and this is the flower itself. So flowers are the plant reproductive organs. Next week we'll talk about the animal reproductive organs. But for plants, for seed plants, it's largely the flowers for angiosperms. And that reproductive organ is broken down into 4 main parts that you want to be familiar with. Those are the carpels, the stamens, the petals, and the sepals. One of those right off the bat, you should just know what that is, a petal. Yeah, so 2 of these 4 organs have a reproductive function. They have some gamete within them at some point. The other 2 are sterile. They help in the reproduction process, in pollination, but they don't have any, you know, gametophyte or anything like that within them. They're sterile. And so if we kind of see the basic diagram of a flower, we can see those 4 parts. And we'll kind of come back to this image here in a second, but sometimes it's better to draw it out piece by piece. So I'm going to go back to the board and I'll just do it right here and talk about each of those 4 reproductive structures. So petal, sepal, stamen, and carpel. So the sepal you always see before the flower emerges. It's actually just the protective covering of that flower, and that's the main purpose for it. It's to essentially protect that reproductive organ until it's actually ready, it's mature enough to kind of undergo that process of reproduction. So it's just that first covering, and once the flower blooms, it's kind of off to the side. So they tend to be green in color, kind of just looking like modified leaves, which they are. But there's nothing flashy about them, because at this point, the main goal of the sepal is to be a shell. It's actually to keep animals away. So you don't want bright coloration or anything like that for the most part in these flowering plants. From there, you know, once the reproductive organ is ready, then you want to get your main form of pollination to you. And so for many flowering plants, that relies on, you know, the petals. And those are going to be different colors based on what they attract best. So the blues and reds of most flowers are largely because they're attracting different types of pollinators. Sometimes they look bland too, but they're actually kind of full of patterns in the UV range to attract things like bees and wasps, which can detect them, whereas we can't. So yeah, the petals meant to signal and attract those pollinators. And then from there, it's the actual reproduction. So the stamen we identify as that male reproductive organ, which I'll kind of draw right here. And the stamen is comprised of two main parts, a long end and where essentially the magic happens. So the anther, where the pollen will actually be created and accumulate, and then the filament, which is just a stalk that holds it all together. But yeah, the stamen is essentially all of this, and it's the anther and filament, just made up of two components. And then the carpel is a little bit more complicated. It's got three main components, but there's going to be another vocab term that we associate with the carpel, which kind of complicates things a little more. And there's a lot going on inside. So the carpel can sometimes be drawn like this. It's essentially kind of like a bulb, a long kind of stalk-like structure, and then what I call like a landing pad. And so these are the three components of the carpel. The stigma, the beginning kind of sounds like stick, that's where the pollen is going to stick onto.
Style.
The style, if you've heard of a stylus pen, a long pen, the stylus, so they actually come from the same word. Style is that long path that the pollen is going to travel to. And then what is the last one?
Anther?
Ovaries.
Oh, over.
Ovaries, yeah. So the ovary, this is where kind of other people kind of get stuck as well, because the ovary has stuff inside there too. It has the eggs hiding within there. But the eggs are also kind of encapsulated in what we call, what are those called?
Ovum?
Ovules,
Oh,
yeah.
ovules.
So the way I try to remember these is, okay, if they're all based off of the same word, egg. So the egg is the ovum, or ova for plural. They're encased in an ovule
Inside the ovary.
inside of the ovary. All of these endings we can kind of make sense of. An ova is just the egg itself. Ovule, ending in ule, is the capsule for the egg. An aerie, like a library, is a house of books. Aviary, a house of birds. And ovary is literally the house of the eggs.
That's good.
Yeah, I mean. But yeah, so basically that's how I try to remember those is, yeah, the egg is inside that capsule, which is inside the house of that, of the egg as well.
Nice.
So, yeah, the carpel, stamen, petal, and sepal. But once you know those four parts, you really want to be focusing on those components of the stamen and the carpel.
Oh, wait,
in
can I take a
particular.
picture?
Yeah.
Okay.
Yeah, so I'm just gonna essentially pull up now kind of what I just drew, but I just like doing it piece by piece since this figure doesn't kind of do it that way. Now there's another vocab term that pops up in here. Don't worry too much about the receptacle. The receptacle is literally just the part of the plant that the flower sits on. Yeah, so, but the one that does confuse, and it confuses me occasionally as well, I always have to kind of make sure I'm thinking of the right one, is pistil versus carpel. A carpel is essentially what I described, this one kind of combination of stigma plus style plus ovary. The thing is, some plants have multiple styles, multiple stigmas, and then they fuse at some point. Each one of those kind of unfused components would be called the carpel, and all of it together is called the pistil. So if a plant just looks like this, where we just have the one style, stigma, and ovary, then it's simple. The pistil is the same as the carpel. The only difference lies, this is just more than I said, when you kind of have multiple. These are multiple unfused carpels, simple pistils. They fuse together, forming a compound pistil. So a compound pistil is basically multiple carpels kind of fused together. So carpels are kind of like the building blocks of a pistil. And that's pistil, I at the end, not pistol, like the other thing. So yeah, actually you all have, seen this, these kind of compound pistils before. Hibiscus have compound pistils, and you can actually see it better when you look at the stigma. Sometimes, if it's a simple pistil, it'll just kind of look like a circle. For hibiscuses, it kind of looks like 3 circles kind of fused together. And the same, I think, for lilies as well. Okay, so I talked about a sort of association you want to make relating to micro versus mega. So you're going to see microsporophylls, microsporangia, megasporangia, megasporocytes. There's basically a mega and micro version. Whenever you see mega, just think there's also a micro version as well. And this just plays on this distinction between the male and female sort of trajectory at this point. So plants can have both male and female reproductive organs. We see flowers with stamens and carpels in the same reproductive organ. Some plants actually do have some flowers that are male, some flowers that are female, but many of them have it all combined. So when we think of the male side of things, we want to think micro. When we think of the female, we want to think of mega. And this plays on this difference between what they're ultimately going to make. So the last thing they make, essentially, is that sperm or egg. And just like in animals, in plants, the sperm are orders of magnitude smaller than eggs. And so that's where the terminology of mega being associated with female, micro associated with males comes in. It's just the sperm happen to be smaller than the eggs. So whenever you're thinking of microsporangia, even if you have no idea where you are when you see this vocab term, you should at least associate, okay, micro is the male. I at least know I'm kind of right now in this part of the alternation of generations. You can kind of hopefully situate yourself.
Wait, which part? The top part?
Yeah, the top part here. That's where the whole breaking off into male and female begins. That heterosporous nature of seed plants means that the sporophyte is going to make male and female spores, what will become male and female gametophytes, which lead to male and female gametes, sperm and eggs. And so when we look back at that structure of the stamen, where really all that matters functionally is the anther, the filament just kind of gets the important stuff up there. The anther is essentially where the pollen is going to be made. So the chambers that kind of make these pollen grains are called the microsporangia. They're making the pollen. So these are the pollen sacs. So that there are 2 words, microsporangia, which are the same as pollen sacs. We'll see what the equivalent is for females, but it's actually pretty similar. So microsporangia are the pollen sacs. They make the pollen, which are that male they're going to house that male gametophyte. We don't have to worry about that part. Okay, so what are the four main structures of a flower?
Petals, stamens, staples.
Yeah. Yeah, exactly. So this is gonna be a bug. Yeah, good. Y'all got it. Yeah. Petal, sepal, carpal, stamen. Yeah. Now I know. Okay, I'm gonna rephrase this question next time. Maybe I'll make an iClicker. Okay. Speaking of, another iClicker question. So these represent the entire male reproductive organ, which contain blank that hold the gametophyte. Blank represent the entire male reproductive organ with contain blank that hold the male gametophyte. Give y'all maybe about 10 more seconds here. Finalize your answers. And okay, let's see. All right, between C and D. Okay, and I can see why you might choose D.
Oh no.
But the correct answer here is C. So it is the entire male reproductive organ. I did say anthers are really what's important, but that filament is crucial as well in that, without the anther up there, the pollen is just going to stay stuck at the bottom of that flower. And they contain those pollen grains, which hold the gametophyte. So the pollen grain itself is kind of like the vehicle that the gametophyte is in. And we'll see just how tiny it is. So remember, we began this by saying that in flowering plants, the gametophyte stage is really reduced at this point. And yeah, so what we're going to focus on in the last portion of class today is essentially getting from the development, the creation of a gametophyte, how it leads to kind of pollination, what happens during pollination, what is double fertilization, and the seed development is starting to get at fruits, and so, you do want to be aware of it, but if there's one piece of info you want to kind of drop out, you really want to prioritize these first three. So don't worry as much about the seed development. Make sure you have gametophyte development, pollination, and double fertilization down. And so, I'll just breeze through this because this is a simple figure and move on. No, this is obviously pretty complex. And yeah, we're going to kind of walk through this, parts of this, kind of what I feel might be the most important components here. But when I say we're talking about gametophyte development, remember that when we think of gametophyte of the flowering plant, that could mean one of two things depending on which adventure you chose, whether you went kind of the male route or the female route. And so the male and female gametophytes develop a bit differently. And so that's really the heavy details in terms of this alternation of generations. So when you see this complicated alternation of generations described here, it's really just, the majority of this is this phase between meiosis and fertilization, where, the other half of it is really just, it goes to a seed, it becomes a plant, and that plant makes a flower. That's really all your textbook is saying at this point. All the heavy details are in that upper half there, in that gametophyte development. And so, for the sake of time, I'm going to post these videos on Blackboard because this one is a bit longer. And what I'd rather do is kind of more slowly walk through the gametophyte development. This kind of sums it all up, so you can have that resource after lecture, but let's kind of go through it more slowly first. So what is the gametophyte? So the gametophyte is highly reduced in those flowering plants. In fact, we'll see it's only a couple of cells in the case of the males, or I think it gets up to 8 cells in females. So it's really only just a small bundle of cells surrounded by some aspect of the sporophyte. And so first we'll kind of look at the female side of things, the development of a female gametophyte. This is called an embryo sac. It develops within each ovule. So if you remember that carpel within the ovary, there's multiple ovules. So this is what's happening, when we zoom in to that ovary. Already a small part of the plant. There's a whole other part of the plant with other flowers going on. And all the gametophyte stages from the female perspective is essentially happening within each of these circles, within these ovules themselves. So from the female trajectory there, this is the gametophyte stage inside of the ovule. Everything else is sporophyte. So that alternation of generations, it's super kind of skewed, right? If it were based on number of cells, it'd be like nothing, essentially. And so, this is essentially called the embryo sac within the ovule. So it forms inside a tissue called the megasporangium. So the megasporangium is that organ that makes a spore. Mega, we know, associate with female. It's the organ that's going to make the female spore. So it's the literal beginning of this part of the alternation of generations. So that's the embryo sac. We briefly saw a couple minutes ago the mention of a pollen sac. So you can probably, we'll come back to it, but you can get an idea that that's going to be maybe the microsporangium. But yeah, the megasporangium is that embryo sac. And then, your textbook talks a lot about integuments that surround the megasporangium. Whenever this is brought up in the textbook, the vocab term you want to kind of more focus on is this idea of a micropyle, which we'll kind of come back to. But all it is, so remember, this is the house of the egg. This is that capsule. If you're the sperm, you have to kind of get in here at some point. The micropyle is just the literal tiny door that allows the sperm in. So this gap is just the micropyle. And it's housed there. So the embryo sac is essentially entirely covered except at that micropyle, at that door. And so what happens inside of here, inside the embryo sac? Well, basically what will happen is you're going to have a cell in here that starts off diploid. I'm running out of whiteboard space, but say we're asking what happens here. Start off with a diploid cell, 2N, and this is the key. This is what it's going to undergo is meiosis. So it'll kind of undergo this process where essentially you can pretty much forget this part, because what it does is it's actually going to become 4 haploid cells. So it kind of undergoes meiosis and then mitosis. And what happens is you've got 4 haploid cells, and then one of them survives. And so that becomes that megaspore. It's going to be the only one that survives. And this is that transition from diploid to haploid. So this is that line being crossed right there. It's just happening in one cell at this point. And so what will happen is you've got this megaspore.
And it divides 3 times.
What's that?
Does it divide 3 times?
Yeah, it's going to divide, go through 3 rounds of division to produce 8 haploid, not cells. It just divides kind of its DNA, so it stays one cell with 8 haploid nuclei at first. So we've got, this N here essentially becomes kind of, if we pause this right here, this megaspore is going to become like 1, 2, 3, 4, 5, 6, 7, 8. They're going to be kind of these 8 haploid nuclei kind of floating around in a megacell here. And we call that the megaspore. So this is essentially that process that gets us right here for the female trajectory. Bloop. And then we're going to develop into that female gametophyte. So what is the female gametophyte? Well, it's essentially just this, what's going to happen next is these nuclei are going to start kind of moving, and they're going to become cells.
Oh, wait,
Basically, cell walls are going to form here. Yeah.
That IRAC question. Okay.
Megaspore,
I
it should
get
have been.
it. No, I get it. So it should have been, for that to be correct, that answer, it should have been megaspore.
Yeah, exactly. Yeah. So megaspore would have been the correct way to phrase that question. But it was megasporocyte, which is different. But yeah, what will happen is kind of cell walls will start to kind of form around here. Oh, did this badly, huh? I'm putting these in specific locations for a reason. And you'll end up with kind of, now, instead of a sort of 8 haploid nucleus single cell, you're now 7 cells. 7 cells with 8 haploid nuclei, which is a little weird because one of these has 2 haploid nuclei just to itself, but something specific is going to happen to this one. These 3 at the top form what are called antipodal cells. Nobody actually knows what they do. So you don't even really have to worry about those. They just kind of they do something else. Then what will happen is this will form what's called the endosperm eventually. This is going to develop into the food for the developing zygote once it gets fertilized. This is a megagametophyte, so something has to become a female gamete. Something has to become the egg, and that's this middle one right here at the bottom. This is eventually going to become an egg. This will become endosperm, but something needs to happen before it becomes endosperm, so it's endosperm to be. And then there are these 2, which are called synergid cells. Basically, they're like the wingman for this egg. They kind of attract the pollen, kind of calling out to it, releasing chemicals that the pollen is sensitive to. And they're like, hey, we got an egg right here. So they're constantly calling to a pollen that is on its way to fertilize. So these are called synergid. They work in synergy. There are 2 of them, synergid cells. And then one that will become the egg. So they're all kind of doing something. This endosperm one is kind of unique at this point because it's the only one with 2 haploid nuclei. And we call those polar nuclei, just because when you look at it under a microscope, they're kind of on either end of each other. But it's 2 bunches of nuclei in one cell. Oh. Okay, so this is kind of highlighted in that video that I'm going to post on Blackboard, but I just wanted to walk through it slowly first because although it's a long video, it goes through this part like just throwing terminology at you. So hopefully after going through it slowly, you can kind of keep up a little bit better that way. So that's the trajectory there for the female gametophyte. We've made a female gametophyte here. All that's waiting now is, that egg, that dedicated egg cell needs to get fertilized. Now we just got to make the male side. So it's going to essentially, instead of forming a megaspore, we're going to have a diploid cell undergo meiosis to make a microspore instead of a megaspore. This one is much easier. Instead of kind of going through this weird set of meiosis and then, choose one, kill the other three, divide. With the microspore, males in the plant world are simpler. It's a diploid cell, just undergoes meiosis once, and there's two cells. One cell does one thing, the other cell does another thing. So it's very easy. Whereas this happens within the embryo sac, in the microspore case, we're talking about the pollen sac. So that is the microsporangia. Embryo sac is the megasporangia. But we actually saw these terms of sporangia before, way back in, I think, protists and fungi. When we were talking about fungi, we talked about how sporangia are the house of the spore. The organ of the spore. It's the same thing here. This is the organ of the microspore, the pollen sac. And all it will do is just undergo, meiosis, forming two microspores, and then they undergo mitosis to produce one generative cell and one tube cell. So all that means is the generative cell is going to be what fertilizes the egg. And then the generative cell has essentially, I don't know football at all, but the big guy who's kind of blocking, I'm actually just going to stop there because I don't know anything about football. A linebacker? I don't know.
Lineman?
I don't know. Yes,
That guy.
that guy. So the tube cell, though, basically, it's what its goal is to do is once a pollen grain. So a pollen grain just has these two cells inside of it, the generative cell and the tube cell. Once it lands on a stigma, the tube cell kind of gets to work, and it's kind of digging. And it's just constantly digging through that female reproductive organ. So through the stigma, all the way down the style. And what it has to actually do is burrow. It can't even go down this way. It's actually more difficult. It actually kind of makes a path, digging this tunnel until it finally connects to that micropile. So the tube cell just does all that work. And then once it gets there, it's kind of out of energy, and the generative cell kind of walks through down a red carpet, and he says, I'll take it from here, and he fertilizes that egg cell. So the generative cell is what fertilizes. The tube cell is literally making what we call the pollen tube. So it's kind of crazy to see when you see under a microscope. You can kind of see these pollen tubes form, and it's crazy to think that just a single cell is actually literally kind of carving away like a Diglett in Pokemon. So, this is essentially.
Or Dig Dug.
Yeah, Dig Dug, yeah. Yeah, so I mean, this is essentially the start of pollination, when the pollen literally lands on the stigma, and then fertilization is going to happen next. And in the last couple of minutes, I'll just bring up this last point. What is fertilization in a plant? So we've done all of this now. We've seen that when we're talking about the female gametophyte, we're talking about making that sort of 7-cell with the 2 polar nuclei there, the synergid cells, and then finally 1 egg cell. That's the female gametophyte. The microgametophyte, which is just 2 cells, 1 generative cell, 1 tube cell, inside of a pollen grain. Now we need to kind of complete this cycle. So that generative cell will also just call the sperm cell. They're going to need to fertilize at some point. And they actually do a double fertilization. So, it was, a little to say that the tube cell does nothing else. It actually kind of does even more. So there are two cells within that male gametophyte. The tube cell digs the whole way there. And then what it's going to do is it's going to actually fertilize that big weird cell with the two polar haploid nuclei. And so when the tube cell does this, it's adding its DNA to this mix. And so you actually end up now with one cell that has 3 bunches of nuclei. And so this creates something we've never actually really talked about in this class. We always think of haploid and diploid, but now we're talking about 3 batches of DNA. And so this is actually a triploid. It's kind of the only time we see this 3N. And this is what becomes that endosperm. So basically the tube cell, on top of doing all the work for that sperm cell, that generative cell, it's now going to fertilize this kind of this cell with the 2 polar nuclei and now become the food for that sperm cell and egg cell. The endosperm is just the food for that fertilized egg. So the generative cell is going to fertilize the egg. The tube cell is going to fertilize that diploid cell, becoming a triploid endosperm. And this is what we mean by double fertilization. So one sperm fertilizes the egg, the other cell, that tube cell, fertilizes and becomes the endosperm. So let's just end things on one eye clicker, and then I promise I'll let y'all go. And it's a banger of a question, but let's kind of go through this. So blank are created within the blank of the male reproductive organs, while blank are created within the blank of the female reproductive organs. Blank are created within the blank of the male reproductive organs, while blank are created within the blank of the female reproductive organs. All right, give you about 10 more seconds since we're at 4:15. Finalize your answers, and bye-bye. There we go. Yeah, y'all are all wrong. No. Yeah, microspores are created within the pollen sacs of the male reproductive organs, while megaspores are created within the embryo sacs. So the embryo sac is also referred to as the megasporangium. The pollen sac is also referred to as the microsporangium. Remember from fungi, sporangium kind of has the letters for organ within it. So the megasporangium is the organ that makes the megaspore. The microsporangium is the organ that makes the microspore. And then that's it. It's a lot to teach too.